It's not some distant, faraway land where papers get published and then 20 years down the line we get a drug.
Dr. Priscilla Chan is co-founder of Biohub. 10 years ago, Dr. Chan and her husband Mark Zuckerberg launched their audacious mission to cure or prevent all disease.
The science has moved incredibly quickly because the patients are bringing assets to the table.
In this fireside chat, Dr. Chan speaks about how her time as a pediatrician shaped her vision today, what Biohub is doing to try to compress decades of scientific discovery into months, and how technologies like the virtual cell and AI might transform both research and clinical practice.
We're fully committed to building tools faster, more effective, and more efficient.
Her vision for the future of medicine may sound like science fiction, but with the power of AI, their team believes that soon enough, it will be very real daily practice. Welcome to Stanford Department of Medicine's Inside Look at the Future of Medicine.
We are very excited to welcome you to Stanford. Thanks for being here.
Thanks for having me.
When we think about the philanthropy and the work you've done through CZI and Biohub, it seems, I think, to flow from your identity as a physician. And I mentioned your pediatrics residency. You've talked in the past about how you remember times when people would show up with really very little knowledge about the rare condition that their kid would have and wanting to really try and help those kids. Talk a little bit about your inspiration for the work that you're doing with CZI, and we'll get onto some of the newer things relating to Biohub shortly.
In pediatrics at UCSF, we saw kids from all over come. We just didn't know what they had. And oftentimes, you get a printed PDF or you're trying to search on PubMed a name of a gene or maybe a bunch of symptoms, and you just, there's not very much information available, and there's definitely not a clear way to translate that to what you're supposed to do when they're hospitalized with you. That was my first taste of oh my goodness, this is where basic science needs to come in and is the source of hope. Because I will tell you, when I was applying to residency, a beloved mentor of mine was like, "You're not gonna go down the basic science route." And I was like, "Oh, okay." I was like, whatever it takes to get into residency, right? And so I sort of went down a different route around leadership and service, but it was those moments when I was like, this is all we have, and this is all that this family is clinging to and in search of answers for their child. So when we had the chance at CZI to work with rare patient groups, I was like, I remember exactly what this was like. And in 2019, we started the program where we gave some seed funding to patient advocacy groups. And at first I thought, okay, my imagination had not been open to what they were going to do. I was imagining sort of patient support, sort of walking each other down the route of diagnosis and treatment options, awareness. But what actually happened was phenomenal. And now we've done multiple cycles of the Rare as One, where we give funding to patient rare disease groups. And they have since taken that funding to convert it into research agendas for their disease. 50 groups working on rare diseases, about 20,000 researchers now plugged into those research agendas. And the science has moved incredibly quickly because the patients are bringing assets to the table. Incredibly important assets without which the research would not happen. They've built disease models. They've helped cultivate cell lines. They've hundreds of cell lines, hundreds of disease models, then clinical registries, biobanks, clinical studies, natural history studies. All of that makes it easy for folks who have then the skills in basic science to then contribute and actually move their science forward that will make a difference for their disease. One that I'm particularly excited about this week is around the FOXG1 mutation. Causes a rare neurodevelopmental disorder in children. And the group just got FDA approval for their clinical study for gene therapy. And I can't even say this without getting a little teary. And the mom who started the group, the rare disease group, promised her daughter they'd have a clinical study by the time the child was 10. And they got it approved on Amara's 10th birthday. That is incredible. It's also incredibly fast. So it's that work has been incredible and inspiring to see how you can do science differently.
Yeah, it's really just incredible to see what these rare disease communities can do. As you know, we've been involved here through our Undiagnosed Diseases Network, which you have supported, and thank you for that, for many years with the rare disease community. But I think until you really look straight in the eye a rare disease parent, a rare disease family, and understand where they're coming from, they talk about being on an island often before they're diagnosed, and then joining a community when they find that diagnosis, and how that's so empowering. When I think of that and compare it to what we often get from reviewers, which is where's the actionability in your diagnosis, the parents of these kids never ask where's the actionability. To them, it's incredibly empowering. That's the start of their mission. And then they come together, they bring together groups like the FOXG1 group that you mentioned, and they are focused on a cure, and there is no one with more energy and motivation and focus than a rare disease
Totally.
parent.
The parents are so powerful and often selfless, because oftentimes this is not going to help their child. And it's about making it better for everyone else.
Yeah, because it can sometimes be too late by the time their action has led to big changes. But no, I think the rare disease community, we're so grateful that you in particular, and obviously the CZI itself, backed them, and in this unusual way, which was to combine support for bringing the parents together and the families together with connecting them to the right scientists, because that's the magic. The families on their own can support each other, but the cures are going to come from a connection to science. Yeah. And science has really been obviously at the center of everything that you've done, and the huge ambition that you have. I wanted to jump up really quite sort of up to date in some of the newer things, because you've announced some really exciting new things just this year, and I was lucky enough to be at your launch for Biohub and with some of the new focus. You've talked about the virtual cell, which may be the hottest thing in biology right now, and there are many groups focused on it. Give us a sense of the scale of that project and why you think that could be the answer to many of the questions that we're trying to answer in basic science and translational medicine.
First of all, virtual cell is super exciting, and there's not even a true consensus of what we're all talking about, so maybe I'll say what it means for us at CZI. We want to be able to build a virtual model that helps us understand what the underlying biology that powers the human cell. So we want to be able to have a computational model that allows us to quickly and more cheaply look at cause and effect in the cell, look at various disease states, perturbations, how various genes contribute to changes within the cell that may later cause downstream disease. That work is super exciting, I think, for medicine in general, and you work in precision medicine, we call it frontier medicine. I think it's this very similar things. To cure, prevent, and manage all disease, that is a big mandate that we don't think we're going to do alone. And the reason why we think we have a good chance at it is because at CZI, we're fully committed to building tools that makes every other scientist faster, more effective, and more efficient at their work and research. If we can speed everyone up, then science just moves faster, and we can have new discoveries, new knowledge that impacts patients' lives.
And to clarify for everyone, maybe some who hadn't realized, that is your mission, to
that
cure, prevent,
yes,
or manage all disease.
We dropped manage. We're
Okay.
we're just going to cure. Prevent.
Okay, that seems fine too. That's not a small ambition. We love that ambition in this audience.
It was very, I will tell you, most reasonable scientists could not look us in the face and with a straight face and say yeah, that sounds like a good idea 10 years ago. And, but we've gotten lucky because right now we're at a moment where I think it actually is feasible and possible. And virtual cell model is going to play a large role in this. Right now, a lot of biology is done, basic science is done through sort of someone having a clever idea, a happy accident, years of dedicated trial and error. And what we're hoping to do with the virtual cell model is actually de-risk a lot of that. Have a computational model that allows scientists to test a lot of ideas, especially riskier ideas, to then say, okay, these are the few that we think are most important and highest yield, and then we'll test it in the wet lab or in an animal or whatnot. But actually, is the holy grail for me? Developing a model that allows us to understand individuals' biology. You talked about the diagnostic journey, which is terrible, and you guys have walked through the diagnostic journey with patients many times. We have fewer answers than questions when we're walking patients through that journey. And it's actually sometimes a relief and lucky when someone gets a rare disease diagnosis. And more often than not, you say, like, I don't know, we looked at your whole genome and we think you have these 3 funny things. And we don't really know what that means. And we don't know what risk profile that that we need to put you into. That is such a tricky part of the patient journey and your journey, walking through with them as the physician. I want us to be able to have virtual models that looks at an individual's biology and says, based on these 3 mutations, we see that that impacts this protein downstream, and that protein is integral in this process, and we can look at the risk profile, and we can also look at treatment, and then can we predict natural history for something that we have no word for right now. We just have a constellation of symptoms and a few genes that we don't know how it actually impacts. That model will truly get us to frontier biology, precision medicine, in a way that allows us to treat the individual that walks into the clinic and really closes the gap between basic science, clinical research, and treatment that I think is super exciting.
Yeah, no, I mean, clearly the cell is the individual unit of biology, right? It's the singular unit. And we have got better and better understanding that. We have single cell genomics, single cell proteomics. We're increasingly able to measure things at a single cell resolution. But I think the scope and ambition of modeling an entire cell, especially a human cell, I mean, perhaps some recent papers are starting to get close to much simpler cells. But to model an entire cell means multidimensional.
Yes.
And then I think part of it as well, and I think you've embraced this already, is also the perturbations. Because it's one thing thinking about a cell, a quiescent cell, just kind of sitting there and trying to predict its gene expression or something similar. But really what you want, as you've been talking, is to be able to predict what happens when you mutate this particular gene in this particular way. And that's another scale again.
And then there's like sort of the spatial aspect, right?
Yeah.
Like things aren't just soup within a cell. You're looking at how things are arranged within the structure of an individual cell. The really exciting thing is maybe, let's say 24 months ago, we're like, are the models powerful enough? Is there compute? We spent a lot of time building up a very robust GPU cluster to allow us to do this type of AI research. But right now, the key constraint is now data and looking at different data formats. There is great data in biology, but most of the data available, whether it's single cell or spatial work or protein work, they are often proprietary data sets, and they're often configured to answer specific questions that someone had gone into this work looking to answer, which is reasonable. But what we really need in biology right now are foundational data sets that are open and standardized and everyone has access to in order to do their research and build upon. And we're very focused on that at the Biohub. We've had some lucky history. 10 years ago when we were told that we were stamp collecting, we started doing single cell, and we built out the Human Cell Atlas, Tabula Sapiens right here at Stanford. And then we built a data set called CELLxGENE, which is one of the largest single cell data sets available. And the incredible thing about those data sets is that they were built with general use and general knowledge. And we learned that no one group is going to build all these data sets. A very happy accident happened with CELLxGENE. We were trying to get, at the same time we were trying to get a single cell data set off the ground, we realized that annotating the data was really hard. So CELLxGENE was not originally meant to be sort of a very large data set. It was an annotation tool that allowed researchers to identify the cells that they wanted to look at. This little tool became very useful for scientists. And because they were all using the same tool, they happened to use the same data format. And then we had data that was building upon each other, and people wanted to then, it was useful, and they started giving their data back. We seeded the original data, we paid for and seeded the original data. And now about 3/4 of the data in CELLxGENE is not something that we sort of put into the world ourselves. People are just building this data set together. And because it's standardized, it is incredibly useful, and it's used by folks across academia and industry. And so we're following that playbook. We are looking to work with academic partners, industry partners to build the next data sets in our Billion Cell project. And we are very careful to look across species, across ancestry and human data, age, disease states. And the key will be to get multiple parties to be excited and willing to put it in the same data format for us to be able to build a foundational asset for everyone to be able to use.
So let me just be clear, you just said 1 billion cells.
I know, it got big real fast.
It got big fast,
Yeah.
right? We were talking about one, and clearly, as you mentioned, over the last decade, you and others, but very big focus for CZI has been single-cell sequencing, as you mentioned, the Human Cell Atlas and all that work, the mouse one as well, and multiple organs. So that was in the range of hundreds of millions of cells, but you've raised the bar a little bit more.
The technique has been established.
Yeah.
It's really about getting the right folks involved, doing the science and bringing it back together. And we're not limited by actually number of cells anymore. It's really like, do we have the right views on the data? Which is so exciting. The world moves so fast. Like, it's so hopeful.
Yeah. I love also that you mentioned the spatial element, because you've also had this interest in imaging.
Yes.
And I feel like I was talking about perturbing the cell, and I feel like our lost dimensions sometimes, like time, is kind of a lost dimension. We stop cells and we kill them and we measure them. Mm-hmm. Whereas in life we get up every day. Time is part of our lives, and certainly we go to see doctors at different points in time. Mm-hmm. And so ultimately, if we go all the way to patients, time is an inevitable dimension. But with cells, we haven't traditionally thought about the spatial element. But you have unique tools that from your imaging Biohub that can speak to that.
I hope that no one here ever needs to go become a cryo-EM slide. Yeah. It's much better to be dynamic, warm, and alive. Both have cryo abilities and our electron microscopes, and we're very proud of the work we've done there. It has been pretty high ROI in investing in a few things, like a laser phase plate in partnership with a group up in Berkeley, to really improve our contrast and resolution through that. So we can see down to the atomic level of what's happening in various cells and preparations. That's really cool. We can actually say, I hope to be able to connect the dots. With this mutation, this protein changes in this configuration, and we can see it clearly with this imaging ability. Next is going to be looking at video, right? We want video. We don't want static slides. So what are the different labeling methods without labeling that allows us to look at living dynamic cells at a resolution that helps us understand how the gene connects to the protein, connects to the living cell?
Yeah, no, it's amazing. And I think it's just remarkable how often the scientists who do that kind of work aren't really talking to the scientists who do the other work. So being able to bring them together, that convening power becomes really important.
It's been awesome. And that's actually the whole premise of our research groups in the Biohubs. We put out a grand challenge, whether it's tissue engineering or cell engineering or single cell work. And we say, this is what we want to do. This is our big pie in the sky. Do you want to do this with us? We don't have
We do, by the way, just for reference.
Yes.
We do, yeah.
It's been great. But, inviting folks from whatever seat they may come from to say, I'm an AI researcher, or, I specialize in the physics of lasers, physicians, engineers coming together and building together as a team and having these multidisciplinary teams be able to do the work towards our grand challenges, be able to answer these questions while building important data sets. In partnership, we have 9 universities, Stanford is one of them, that partner with us deeply. and at the same time build something that's open and available for everyone, far beyond the 9 universities to build upon. That's our big goal.
Yeah, that's amazing. One of the things, though, that of course that happens when you bring people from very different parts of science together is they have slightly different views on how long things are going to take. And I love this.
Yes.
You know, as part of, as you were relaunching or launching Biohub, for the next 10 years, the AI people were on one side saying, biology, oh, we can solve that in, you know, give us 2 or 3 years.
Yeah,
you know.
they got it. It might be 18 months.
Yeah, and the biologists are on the other side going, what? Come on, you know, we've been doing this for decades. How do you parse that? Who do you believe? How do you resolve that? Or you just throw them together and like see what happens?
Well, I'll tell you guys a funny story too. This is not a new problem for us because we have always had a mix of very different people coming together. And Corey Bargman, our founding scientist leading our work at CZI, joined us 10 years ago. I adore her, love her. And we wrote down one day in a team meeting, we have to sequence our priorities. Maybe some people see where this is going. And she writes down, sequence the priorities. And then she looks around and she goes, where will we get the samples from? And I was like, that's not what we're talking about. We just need to write down what we want to do, Corey. So we've always had the excitement of having people from all different backgrounds come together and sort of the additional work that it takes to really get people from different trainings, different perspectives to come together at CZI. Who do I believe in this? Well, I believe no one.
Yep, that's good. That's good. I like the skeptical scientist.
And the cool part is just like having people say, well, this is why I think it's impossible. My favorite are the people who think it's impossible, because the people who think everything is possible, they just haven't gone down far enough, right? But the people who think things are impossible, you're like, enumerate why you think it's impossible. What are the blockers that make it so that you say that? And then there are people in the room who can say, oh, I actually, from a different experience, know how this works. And you know, that's when the problem solving or sort of the resource allocating and figuring out how to solve the problems together happens. I would say I expect science already looks different.
Yeah, completely.
Therapeutics are being identified and built using AI right now. Right. And right now it's anecdotal. You're like, oh, this cool thing happened. But I think maybe in 5 years, we are systematically working in a new way, and the breakthroughs aren't anecdotal. We're really looking at us marching through a deep understanding of human biology and helping patients on a regular basis.
I was wondering a little if Mark was on one side of that and you were on the other, and you were balancing the two, or
the Mark and Priscilla collaboration. Like, what does that look like? Is that what you're asking?
A little bit. Yeah, a little bit.
Where you see Mark is really through the engineer's perspective. Like the idea of like doing the single cell transcriptomics and building a virtual cell model where you understand the system works. Like that very much appeals to him, because he wants to be able to understand and instrument the system, understand where the bugs are, go in, look at cause and effect, and tinker, and say like, here's the system. Yeah. Like if we understand something at the fundamental level, then we can understand human biology. So that very much leans into sort of what he looks for and advocates for. For me, is always the question of are we answering important questions for patients and for clinical use? Yeah. And that's the bit that I'm always like, okay, is this interesting? Like why? Like tell me why building a series of mirrors around the cryo-EM is going to help patient care later down the line. And I think the work that's so exciting is that we have a strategy that really closes the gap between the physics in electron microscopy to the impact in a patient. Like that is cool. Like bringing together basic science and clinical work to be side by side, that is the future of medicine.
Yeah, that's, we couldn't agree more with that. And I think having computation next to, and by the way, you mentioned your cluster. It's the largest non-profit GPU cluster, basically outside of tech. It's the largest GPU cluster.
Yes.
So you're ready to do this, but having that next door to these wet labs, as you're talking about, and the cryo-EM, and then have on top of that, essentially your philosophy of what does this mean for a patient. is I think, what this sort of seems like the North Star of what you're trying to do.
I mean, it's an incredibly exciting time for biology right now.
We talked about cells. You've also have a project called the virtual immune system, which
Yes.
is really interesting. And I think some of the researchers are thinking about essentially immune cells like floating around the body, surveying, looking for disease,
Yep.
extinguishing disease wherever they find it. I mean, I do not underestimate immune cells. They are, T cells are exquisite. We all have our favorite cell.
Yeah. What's yours?
Maybe an NK cell. I have a friend who does a lot of work on that.
Okay.
So she's kind of persuaded me over the years. Yeah. What about you?
Oh, what is my
I
favorite?
mean, I have to say a heart cell actually, but so I should.
I thought you were going to say heart
I should have
cell.
said a cardiomyocyte. So that's obviously the anser. Just forget I said
I
anything
hope
about natural
that your
killer
favorite
cells.
cell is the cardiomyocyte.
It is. Yes. Let me rephrase my answer.
I don't know if I have a favorite cell. I have to say, maybe my favorite cell doesn't exist yet.
Ah, that's a good answer. That is a very good answer. Yes.
So you're talking about our work at our New York Biohub,
Yes,
where we're
yes.
doing cellular engineering. Our goal is to be able to build an underlying platform. I'm using technology platform, not the software kind, where we can help cells be targeted to a specific destination, read out a certain state that we're looking for, encode it back into its own DNA, and lyse itself, so then we can pull out the cell-free DNA and read out the data that it has gathered within the human body. Like that, and then the next step will be like, oh, can you then get it to take an action? So like that would be incredibly cool.
Yeah.
And the example I like to give is about the heart. I like to give the example of like, can you use this technology, send a cell to read about the, how the plaque burden of a coronary artery, encode the data, lyse, read it out, so that, to get that information, you're injecting cells into a patient and then drawing blood and sequencing it, and Obviously, that is still science fiction, hopefully not for long, because we have a lot of unanswered questions about sort of like the inflammation that you would trigger, what would happen, can we actually read with fidelity? But that's the type of technology and platform that we think about building, because that's a general purpose technique. Because then you can do a lot of other things. You can go into very privileged places in the human body. And read out data that's useful for us to know that's minimally invasive. But that's where partnership with the community comes in. We see ourselves as building the platform and technology and in partnerships with others, but we need scientists, physician scientists, to be thinking about the applications of that data, that technology, and how they would use it in their daily work.
Yeah, I mean, that creative process, you used the word science fiction. I was going to use that myself because it seems so futuristic. But I've been thinking about this even just about the last few days, how so much of the world we look around us right now, particularly where AI is concerned, and it looks like science fiction from just a few years ago. If you do your own version of science fiction and look forward 10 or 15 years, and we were talking about the movie in theaters right now, this Project Hail Mary, which sounds like it's really excellent.
It's so cute.
Yeah. But Rocky, that's the name of the alien,
Rocky.
right?
And
okay.
they named the planet actually, I'm not going to say anything.
Well, no spoilers, okay,
Yeah.
but it's a good movie. But I think science fiction is creative people, many of whom understand science, thinking about what the future could be like. But in a way that's kind of what you're doing, and also trying to help build that. If you have succeeded, and you recently looked forward to the next 10 years, given the tools we have today, as well as back on what you'd built, the foundation you built for the last 10 years, how do you see that medicine has changed? Because we're the ones practicing, and we all love science, and lots of us have labs as well. But we are hoping, as you are, I think, with your vision, that we will change how we do medicine.
Yeah, I talked a bit about sort of being able to have that virtual model of individuals. I think that is super powerful to be able to look at sort of individuals' genetics and actually be able to translate those genetics in a meaningful way to clinical practice. I hope very much that we will start looking at individuals like in that way 10, 15 years from now. But I also think in terms, this Stanford University, God bless physician scientists, especially those who've got an MD, PhD. That is so long. I think if I stayed in medicine, I would still be finishing if I was in that route.
And you know that you're always welcome back. We are.
You know, that is actually my secret retirement plan.
Okay.
You know, and I wanted to be a pediatric cardiologist.
Oh, well, now we're talking.
Yes.
Yeah.
And I actually did a rotation here. It was one of those things that maybe Stanford had some asset over UCSF. So I spent some time here. It was really something I wanted to do, and I haven't closed the door on that.
Okay.
But that's a separate, you know, kids have to go to college. And
Yes,
I'll be the
right.
oldest fellow that's ever been.
Okay. But when you're ready, we know some people.
But I keep my board status active because I plan on this.
Okay.
The bit that I think will hopefully, the way physicians can evolve their practice and that we evolve training is that everyone is actually a physician scientist. It's not some distant, faraway land where papers get published and then 20 years down the line we get a drug. I don't want us to operate that way anymore. It is slow and effective. We have better tools than that. Like, how can we actually close the gap? And everyone is thinking deeply about the biology, the protein structure, the mutations that are happening within individuals, and the factors that put them at risk, and use that in our clinical practice every day. That is sort of how we get to that individualized care that we're all looking for. And that is how we can actually very quickly translate what's being learned in a lab to impact patients. And that has to look different in 10 years.
Thank you, Dr. Ashley, as always. And please join me in thanking Dr. Priscilla Chan.
Thank you, everyone.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.